based on the hill to the far right. We've got a great outdoor lifestyle in Exeter, so this is obviously the summer, but lots of paddleboarding, so I do a lot of paddleboarding myself. We've got this beautiful old cathedral as well. We're right on the southwest coast path, so you can go walking the entire length of the UK coast, hiking, which is really, really pretty. And finally, I was actually over here last week actually doing some hiking on Dartmoor, so we've got all these natural tours and natural moors. So really, really pretty place in the UK. It's where most people in the UK go on holiday. So I'm really lucky to actually be a researcher and academic and get to live somewhere like this, because I used to be in London before. So on to some science. So I'm going to talk to you a bit about Alzheimer's disease. So Alzheimer's disease is the most common neurodegenerative disease, most common cause of dementia. The disease itself is caused by two protein hallmarks, so the accumulation of amyloid beta plaques, which are shown on the top left, and they're essentially extracellular plaques within the brain, and then slightly later in the disease you get these neurofibrillary tangles, which are hyperphosphorylated tau protein, and these form within neurons, and these occur slightly later in the brain. At a macroscopic level, you can see quite big differences in the brain of someone with Alzheimer's disease. So on the left is someone without disease, and on the right you've got someone with severe Alzheimer's disease. And you can see that there's considerable shrinkage of the brain by end-stage disease. And actually, it's the entire weight of an orange someone has a loss of brain matter by the end of disease. So there's a lot of atrophy. Within the disease, we use the spread of the tangles throughout the brain as a way to stage disease. So this is called Braak staging, and it's spread of those neurofibrillary tangles throughout the brain. In the initial stages 1 and 2, they're largely located in the transentorhinal regions, and then in the later stages, they spread through the entire cortex via the hippocampus. This is known as Braak staging, and stages 5 and 6 shown at the bottom, we've got really widespread tangle deposition, and this is the criteria used to diagnose Alzheimer's disease post-mortem. In stages 3 and 4, shown on the top right, this is when you start to see clinical symptoms of the disease, and stages 1 and 2 are clinically silent, and it can sometimes be perhaps decades or so between someone actually getting pathology and then starting to develop clinical symptoms, which makes it a real issue to try and treat this disease. So as I said, there's no definitive pre-mortem diagnosis. We can currently only accurately diagnose Alzheimer's disease post-mortem, and the neuropathology precedes clinical symptoms. And because neurons are post-mitotic, it means it's incredibly hard to try and deliver effective therapies for the disease. So we've long known that Alzheimer's disease has a heritable component. So a small proportion of Alzheimer's cases are familial. They're caused by autosomal dominant mutations in APP or presenilin genes, which are part of processing amyloid beta. The rest of Alzheimer's disease cases, so over 95% cases, are sporadic. We still know that genes are important. And this is a twin study, actually from the 1990s, but the statistics are still true. You can see here's Alzheimer's disease, and you can see when you've got identical twins, which are shown in black, you've got about twice as much twin concordance for disease as unidentical twins, which shows genes are important. But it also shows that genes don't explain everything, because we've still got 40% identical twin discordance for disease, which suggests that other, perhaps environmental influences are playing a role. So over the last, I suppose, 10 to 20 years, there's been a lot of genetic research going on into Alzheimer's disease. So big genome-wide association studies have identified, using maybe 1 million or so samples, have identified common disease variants that can increase your risk of developing Alzheimer's disease, but only by a very small amount. But collectively, these can contribute to disease. And we think that common single nucleotide polymorphisms explain about a third of disease incidence. We also know that environment's important, and some of the recent studies in the last 5 to 10 years have also shown that a number of environmental and lifestyle factors, such as physical exercise, the amount of alcohol you drink, social interactions, and drinking, can also increase your risk of developing Alzheimer's disease. And actually, there was a study that came out from some colleagues in the University of Exeter where they showed that even if you've got a high genetic risk for Alzheimer's disease, so you've got lots of the SNPs that can increase your risk of disease, you can actually reduce your risk of developing Alzheimer's disease with a healthy lifestyle. And conversely, you can actually decrease the converse, so you can also increase your risk of developing disease even if you've got a low genetic score for Alzheimer's disease, if you've got a considerably bad lifestyle. And so basically what we believe is it's not just genetics, but the environment that together lead to people getting dementia, and that we think this is driven by epigenetic mechanisms. So epigenetic mechanisms give rise to phenotypic differences, and they're really important mechanisms that control just normal cellular development. So if you take a number of organs from the same individual, all of these have the same underlying genetic code. They all have very different functions and appearances because of epigenetic differences, which basically turn certain genes on and off in different tissues and at different stages of development and the lifespan. There's a number of different epigenetic mechanisms. The one we're interested in is that of DNA methylation, which is the addition of a methyl group to a cytosine, and this is traditionally thought to bring about gene silencing. However, depending on its location within the gene, recent evidence suggests it can also be associated with increased gene expression. And the way that we look at this is we use the Illumina array, which allows us to look at nearly 900,000 methylation sites within the genome with the current version of this. And essentially what we can do is compare individuals across different stages of disease or compare Alzheimer's disease to controls. So just a bit of an introduction to my team. So I've currently got 7 postdocs and 4 PhD students in my group. And although most of my talk today is on Alzheimer's disease, we're also actually doing work in dementia with Lewy bodies, Parkinson's disease, and Huntington's disease. We're also looking at the effect of different symptoms as well. So for example, I'll talk a bit about a study we're looking at where individuals have psychosis in disease. We're looking at predominantly brain, but we're also looking at blood to try and identify new biomarkers. We've done a lot of work looking at methylation changes, but now we're trying to integrate in gene expression and microRNAs as well to have a more holistic view of what's going wrong at the genetic level in these diseases. And then finally, we're integrating this using different network level analyses. So the first studies I want to talk to you about today were the ones that kind of were the first ones we did, well, 11 years ago now. And we published these back-to-back with another group. And these were the first large-scale epigenome-wide association studies undertaken in Alzheimer's disease. And so essentially what we did was we took 120 individuals. We looked at 4 different brain regions. So the entorhinal cortex, which is affected relatively early, superior temporal gyrus and prefrontal cortex, which are affected in the middle stage of the disease, and the cerebellum, which isn't affected really by pathology. And what we did was we looked for an association of DNA methylation with Braak stage, which I introduced earlier as a measure of tau pathology within the brain. And essentially what we did then was we just looked to see what was our top differentially methylated site we saw in the entorhinal cortex as the region of the brain that's affected earliest. And what we thought was quite interesting was our most significant differentially methylated position, or DMP, was in the ANC1 gene, which is the top one shown in this table and is highlighted in red at the top. And also the 4th most significant gene loci was also in this gene, was located only 91 base pairs away, which we thought was quite interesting. When we look at these 2 loci in a bit more detail, you can see the 2 of them shown there on the top left in the entorhinal cortex. You've got the methylation level shown on the x-axis, on the y-axis, and Braak stage, so measure of disease stage on the x-axis. And you can see with increased disease stage, we see increased amounts of DNA methylation at both sites. We also see this in the other 2 brain regions we looked at. So in green, you've got the superior temporal gyrus, and in red, you've got the prefrontal cortex, which affected slightly later in disease, and we can see the same pattern. And what's really nice is we don't see this at all in the cerebellum, so it seems to be cortex-specific where we're seeing pathology. As these were the first EWASs to be performed in Alzheimer's disease, and actually some of the first EWASs to be performed in any complex disease of the brain, we had to go on then and replicate this. So shown on the top left is where we took the 100 most significant loci we'd identified in our cohort and correlated the effect size, so the change in methylation between someone with severe Alzheimer's disease and control in our discovery cohort, which is shown on the x-axis, and a validation cohort shown on the y-axis. So the top one is an independent cohort of 150 brain samples from Mount Sinai, and we can see a really nice correlation of the effect size with the 2 ANC1 probes shown in red. And then at the bottom, this is actually the study that was published back-to-back with ours. This is another independent 780 prefrontal cortex brain samples. And again, you can see a really nice correlation of the effect size. 2 ANC1 probes showing the same direction too. As the arrays were relatively new at the time, we also wanted to go on and validate this using another technology. So for this, we use something called pyrosequencing, which allows us to look at zoom in on a region of the genome and look at it in a bit more detail. So this is another independent cohort. So this is 40 Alzheimer's disease and 20 control samples, and we used a method called pyrosequencing, which allows us to look at those 2 ANK1 probes shown in red that are 91 base pairs away from each other, but also another 6 sites within that region. And you can see consistently across that region, we're seeing increased DNA methylation in Alzheimer's disease, or Braak stage 5 to 6, compared to controls, which are 0 to 2. And we can sum this as an amplicon average, which is shown at the bottom. And you see in all 3 brain regions, so entorhinal cortex in blue, superior temporal gyrus in green, and prefrontal cortex in red, we're seeing consistently increased DNA methylation in this region. And I think this is really neat because this is quite a small sample size, and we're still seeing consistent changes. What we then went on to do, this is a study that was published a few years later in 2019, and this is where we decided to use our pyrosequencing assay to look at ANK1 and see if it's specific to Alzheimer's disease or whether this is common to other neurodegenerative diseases. And what you can see is in the case of vascular dementia and to a large extent Parkinson's disease, don't really see any difference between diseased individuals and control across the region. Interestingly, we do see a change in Huntington's disease across the region, and we do see a change in dementia with Lewy bodies. But what we do know is that individuals with vascular dementia or dementia with Lewy bodies rarely have pure pathology. They also have often coexisting Alzheimer's pathology. So when we stratified our samples by whether they had coexisting Alzheimer's pathology, we actually saw that now a change in the vascular dementia samples, but also we saw that the change we're seeing in dementia Lewy bodies is driven by the coexisting AD pathology. So this ANK1 methylation change seems to be specific to Alzheimer's disease and to Huntington's disease and is only seen in the cortex. So what I've been presenting so far, I've been talking about DNA methylation, but I've kind of been skimming over the fact that actually the methylation that I'm showing is actually a sum of 2 modifications, so DNA methylation and DNA hydroxymethylation, which are the 2 modifications on the way back to unmodified cytosine. And what's recently been shown is that 5-hydroxymethylcytosine, which was originally thought to be just a passive intermediate in this demethylation pathway, is actually enriched in neuronal genes and is at higher levels in the brain compared to other tissues. So we thought perhaps actually what we might be detecting is DNA hydroxymethylation rather than DNA methylation. Now there's an adaptation to the protocol we use for measuring DNA methylation, which essentially allows us to look at DNA methylation and DNA hydroxymethylation in isolation. And so we use this for our pyrosequencing assay. So this is another independent set of samples. And this is where we've used the standard assay, which allows us to look at both modifications. And you can see pretty much what I've shown you before, an increase of about 5% DNA modifications across the region in Alzheimer's disease compared to control. When we actually try and separate out and stratify by whether this is driven by methylation or hydroxymethylation, we can actually see that there's about a 10% change in DNA methylation across the region. And this is because we're having confounding by DNA hydroxymethylation, which is showing a 5% decrease across the region, and which goes to show that actually in neurodegenerative disease research, we should also not just think about DNA methylation, but we also need to think about DNA hydroxymethylation as well. So following on from our initial EWASs where we focused on ANK1, we later went on to publish in 2018 another EWAS, but for this one we decided rather than focusing on individual differentially methylated positions in the genome, what we do was try and identify differentially methylated regions consisting of multiple adjacent methylated sites, which might suggest something more biologically meaningful is happening in the region. So for this, we had a cohort of around 146 samples, separate from the discovery cohort in the first study I talked about. And for this, this is shown for the prefrontal cortex here. And this is a Manhattan plot, so showing the association with DNA methylation with Braak stage across the region. And you can see we've got a number of genome-wide significant loci, which are the ones shown above the red line. What's quite interesting is we're seeing this nice peak of HOXA3 on chromosome 7. And actually, when we went on to do regional analysis, which allowed us to cluster together these, we found around 21 genome-wide significant differentially methylated regions. And actually, of these, 7 of them were in the HOXA3 region. And this is just showing the top one. And you can see across the, I'm trying to add these up now, 8, I think, individual loci within this region, we see consistently increased DNA methylation in the Braak stage 6 samples, which are the AD, which is shown in grey, compared to the Braak stage 0, which are control. This is just zooming in on that region in a bit more detail. So this is a zoomed in Manhattan plot. And for this, we've color-coded it. So increased DNA methylation is shown in red, decreased DNA methylation is shown in green. And you can see this very defined region of increased DNA methylation, which not all of it's reaching genome-wide significance, but it's showing some kind of common change. So we went on to try and validate this. So this is taking our discovery cohort that I talked about earlier, and although it's not reaching genome-wide significance, you can see that same pattern, increased DNA methylation, spanning around 50 kb of the genome, which is shown here. And this is correlating the effect size. You can see a really nice correlation of the effect size of these probes. The second validation cohort, exactly the same thing, a very defined pattern of increased DNA methylation across the region of 50 kb. And again, a nice correlation. And then finally, when we try and meta-analyze these, we can actually see this very clear region with a lot of the sites actually passing genome-wide significance. So there's now been a growing number of EWOSs in AD, so we've published quite a few of them, but collaborators around the world have published a few more. And what we were starting to see was consistently the same genes were starting to come out, which is really good. But we're seeing ANK1 come up by other people, HBDF2, and for example, HOXA3, which I just talked about then. So we wondered, could we actually meta-analyze AD EWOS studies? We know this has been successfully performed in the GWAS field, and like I said earlier, there's now, I think the latest meta-analysis of AD GWAS is over 1 million samples. But it's not as simple for EWOS for a number of reasons. First of all, as I said earlier, DNA methylation is cell type and tissue type specific. Another issue is that the EWOSs have been formed in different brain regions and slightly different tissue types. Some studies, such as our first one, have used multiple tissues from the same individual, which can considerably inflate and bias your data. We know that many probes are highly correlated with adjacent probes. They're not independent tests. There's differences between studies, so Braak stage is okay, but if we're looking at clinical diagnosis, this can differ slightly between different cohorts. And there's also, we noticed, inter-study differences in sample and data quality. So, for example, some people, like ourselves, might completely randomize our samples when we run them, but other people, for example, because they're looking at something different, might not have randomized them in exactly the same way. So this was a study that we started, I think it was 2017, and it was going to be a really big study, and it took us about 4 years just to work out the bioinformatics for it. It was probably the biggest scientific headache we've ever had was trying to get this working, but we've done it and it's published now. And what we worked out was it really requires really careful data harmonization, really stringent quality control, dropping samples we weren't sure about, and accounting for correlation structure. So this is the study we published a few years ago. And essentially what we did was we performed a meta-analysis combining together every single cohort we could access, both by ourselves and by collaborators, where there were more than 50 samples available. We looked at the prefrontal cortex, where we had access to 961 samples, the temporal gyrus, where we had access to 608 samples, and the entorhinal cortex, where we had access to 188 samples, and the cerebellum, which I'll mention in a minute. So first of all, we performed individual within tissue meta-analyses, and we saw the usual suspects. So there's HOXA3 on chromosome 7. You see the nice peak on prefrontal cortex and temporal gyrus. Interestingly, it's not seen at all in the entorhinal cortex. We've gone back and actually looked at this in all the individual cohorts, and it's never showing increased DNA methylation there, which is probably why it would be why we didn't identify it in that first study, but it shows that's a really interesting gene, because actually it's a neurodevelopmental gene, and we're not seeing it in, for example, the entorhinal cortex. So some of the loci we're identifying aren't across the entire cortex. There's ANK1 shown there, which we see across all 3 brain regions, CDH23, RPL30, and RHBDF2, which were the common ones that were coming out of the literature. We see some uncommon loci across the cortex, like ANK1, and like I said, some are only seen in some tissues, like HOXA3. But really interestingly, the cerebellum, which we then went on to look at, so this is where we've got 533 individuals, so we're absolutely adequately powered to find differences. We see absolutely no association across the region, so all of the epigenetic changes we're seeing in Alzheimer's disease are specific to the cortex and aren't seen in the cerebellum. Then we thought, can we try and identify some common changes in the cortex by combining together all of those different tissues we have available to try and maximize our power? And so for this, we then had to perform an inverse variance meta-analysis, which allowed us to control for the matched nature of some samples, but it resulted in nearly 1,500 unique individuals being used in this. And this is a Miami plot, so we're showing increased DNA methylation above the x-axis and decreased DNA methylation shown below the x-axis. And what we identified was 220 genome-wide significant loci. The majority of these were hypermethylated, and actually only a small number were hypomethylated. There was a significant enrichment for hypermethylation. We then went on to try and validate this in some other cohorts. So the one shown on the left is a cohort we hadn't used because it had fewer than 50 samples available. But what's quite nice is they'd actually sorted neurons and glia, so it allowed us to look at effects there. Again, we saw a nice correlation of the effect size. And the one shown on the right used a different version of the array to what we'd used, and so to try and reduce any variance caused by technical issues, we hadn't used it in our meta-analysis. And so this was another, I think, around 500 samples shown here. But you can see, even though it's a different version of the array, we actually see in an independent sample a correlation of the effect size of these 220 genome-wide significant loci. This is just showing my favourite ANK1 gene in a bit more detail, but you can see here on this forest plot, in the prefrontal cortex shown in red, temporal gyrus shown in green, and entorhinal cortex shown in blue. You can see across each individual cohort, and then the meta-analysis, which is the diamond, you can see consistently increased DNA methylation in disease compared to control, which isn't seen in the cerebellum, which is shown in orange. And at the bottom in purple is that small cohort that I mentioned. And what you can see is that, sorry, the text might be a bit small, but in their frontal cortex samples, we're seeing the same direction as we're seeing in the other tissues, so we're seeing the same thing. We're also seeing this in the neuronal negative proportion they had, so the glia, but we're not seeing it in the neuronal positive samples, which are the neurons, so it seems to be driven by glia. And actually, in a more recent paper that we've published, we've actually gone on to look at this in a bit more detail, and this shows for the 220 loci we identified which cell types they're enriched in. So for this, what we did was fluorescence-activated nuclei sorting, and then performed methylation arrays on these. And then what we could see was for the 220 loci, where we're seeing the disease changes, it seems to be entirely driven by glia and not neurons. And the other thing we went on to do was try and see if we could try and use the methylation changes we identified to try and develop a classifier which could say how much pathology was in the region. Obviously, this would have absolutely no clinical utility because it would only work on post-mortem brain samples, but it tells us a bit about how robust the changes we're identifying are. And so we saw when we performed this classifier that in both the initial discovery samples, but in those two validation cohorts as well, we saw about 80% of classification of a sample as to whether it had high amounts or low amounts of pathology. So these are really robust changes we're seeing, and it explained about 20% of the variance in samples, which is really neat too. So following on from this, we've started to expand our work to not just look at Alzheimer's disease, but to start to think about other symptoms and issues with Alzheimer's disease. So we've recently completed an EWAS where we've looked at whether people had an infection at the time of death with Alzheimer's disease, because we know that actually that can progress the disease quicker. We've also just completed an EWAS where we've had access to a unique cohort of samples where they had immunotherapy during life, and actually we can then do an EWAS about the effects of immunotherapy. The one that I was just going to mention today is one that we got funded by the NIH, which was to look at signatures of psychosis in Alzheimer's disease. So psychosis is quite a common thing we see in people with Alzheimer's disease. You see it maybe in about 50% of individuals. And people used to initially think of it as, you know, just a symptom of some people with Alzheimer's disease. But there's now evidence that it might be a slightly different disease course. And people with psychosis actually are more likely to be institutionalized and die far quicker from the disease. So it might actually be a subtle endophenotype of Alzheimer's disease that we wanted to look at in a bit more detail. Where I say psychosis, I'm not talking about your typical psychotic symptoms that you'd associate with schizophrenia. These are more subtle symptoms. So, for example, people thinking that their spouse is having an affair or that their carer is stealing from them. And these kind of small delusions and hallucinations actually we classify as psychosis and can actually mean that you've got a slightly different disease course. One of the issues is people with psychosis are often given antipsychotics, which can actually make their Alzheimer's disease worse. So we really do need to identify better targets for treating psychosis, which is where this study came about. So this study, well, actually, I'm going through the proofs at the moment, so it should be online imminently. So for this, we looked at two different cohorts. So the Pittsburgh ADRC samples, which is a cohort of samples collected in America, where they'd had really detailed clinical phenotyping and neuropsychiatric inventories performed during life, and the BDR cohort, which was a similar cohort in the UK, but it didn't have quite the same level of detail. So we used the Pittsburgh ADRC samples for discovery and the BDR samples for validation. And for this approach, we used something slightly different. So we decided to use weighted gene correlation network analysis. And so what we're assuming here is that genes within a common pathway, which might have a biological effect, show collective co-methylation changes. So this method essentially allows us to identify modules or clusters of co-methylated genes, and then we can associate them with phenotypes of interest, and then look at those modules that are associated with the phenotypes in a bit more detail. This is essentially what's shown here. So the program will assign an arbitrary color, which is the one shown down the left, to each module of co-methylated genes. And then you can then associate them with your phenotype. So you can see in our comparing AD with psychosis without psychosis, we can see there's 5 modules we identified in our discovery cohort that were associated with psychosis, and these are shown on the right, showing the correlation between their gene significance, so how significant each site is within the analysis, and their module membership was shown on the x-axis, so how important they are to a module. So the more integral they are, the more likely they're going to be driving these biological changes. We then went on to try and validate this in a second cohort, so the more relevant graph to look at is, or plot to look at, is the one on the right. And essentially this is where we look at how well preserved those modules are in our independent cohort. So anything above the red line we say is highly preserved, and we can see that 3 of the modules we'd identified, so the dark red, medium purple 3 and the light blue 1 are all preserved in this BDR cohort, which we thought was really interesting. And then we can go on and correlate these modules again with our traits of interest. So even if a module's replicated, it still might not have an association with our phenotype of interest. So then this is the third step in the analysis. And you can see here that one of our modules, which is the dark red, not only replicated and was preserved, but it also was associated with psychosis. And you can see it shown on the top left in the Pittsburgh ADRC and the bottom right for BDR. So we're seeing a correlation of gene significance with module membership. When we do gene ontology analysis on this, we can see an enrichment of synaptic genes, which I suppose is slightly unsurprising, and we're seeing that across all types of biological analysis we're looking at. And then lastly, what we wanted to go on and do was to try and see if we could have any more biological meaning out of our analysis. So for this, we did a process where we basically do co-localization and MQTL analysis. So the rationale for this is there's been lots of GWASs that have identified SNPs that are associated with disease. One of the issues is that a lot of the SNPs are in non-coding region of the genome. We have absolutely no clue what they're doing. If we can, an MQTL is basically a methylation quantitative trait loci where genetic variation affects the methylation of a gene, either in cis in the same gene or in trans in another gene. So if we can identify a methylation change that's associated with an MQTL, and that SNP in the MQTL is the same as a GWAS, that suggests that that GWAS signal is acting via the methylation change we're identifying. And so essentially we did this, we identified MQTLs, and then we co-localized these with GWAS summary statistics for a range of related disorders, so for example, educational attainment, schizophrenia, bipolar, major depression, etc. And what we saw was that we saw co-localization both in cis, so within the gene, and in trans, in other genes, and these are not cross-chromosome, these are all within the same chromosome. And what we're looking at here is the probability. First time I looked at this, when my postdoc sent it to me, I was like, these p-values look terrible. And they were like, Katie, these are probabilities, not p-values. So the larger the number, the better. And so what we're interested in is H3 and H4, which is basically the probability that a single genetic variant is causing the MQTL and the GWAS signal and the methylation, essentially. And what we want is those to be as high as possible. And essentially what we're seeing is that for H3, for example, for schizophrenia, we're seeing a really high, so over 0.999 for the co-localization with a GWAS region that's been identified from a schizophrenia GWAS, which is really neat. And then we've seen this across a range of different related disorders. And the final thing what we did was we wanted to see which cell types might be driving this methylation change. So we took single cell RNA-seq data and then looked at exploring which cell type using that data to basically map to our methylation data using a process called expression-weighted cell type enrichment. And based on this, we can show that our dark red module, which we'd shown was co-localizing with schizophrenia, GWAS signals, and was associated with synaptic processes, was enriched for methylation sites known to be expressed in excitatory neurons, which we thought was really interesting. After looking at Alzheimer's disease predominantly for most of our group's research, in the last 5 or so years, we've also started to expand this look at other diseases, particularly Lewy body diseases. So Lewy body diseases include Parkinson's disease and dementia with Lewy bodies. And these diseases are caused by the accumulation of Lewy bodies, which is alpha-synuclein accumulating in these Lewy bodies within the brain. And essentially, we also stage this as Braak stage, but it's called Lewy body Braak stage. And essentially, you see the spread of Lewy bodies through the brain, starting the substantia nigra and spreading through the brain. One of the things that's quite interesting is people with Parkinson's disease, about 80% of them will go on to develop dementia, and we call this Parkinson's disease dementia. And actually, post-mortem Parkinson's disease dementia and dementia with Lewy bodies look identical. However, people with Parkinson's disease dementia might have had motor problems their whole, like for years and years, and then they start to get memory problems towards the end. But also, you've got people with dementia with Lewy bodies who might never have had any motor problems, but actually can get quite rapid onset of cognitive changes, and you often get a lot of the psychosis that I described earlier in dementia with Lewy bodies as well. So it suggests that there's perhaps slightly different diseases, even if we're seeing different pathological, seeing the same pathological endpoint, we're seeing actually slightly disease processes. So what we want to do is try and compare these. So first of all, in our first study in this area, what we want to do is initially just look at Parkinson's disease, but see if we could try and look at different symptoms within this. So this is a study that's impressing MPJ Parkinson's. And essentially for this, we used our weighted gene correlation network analysis approach, and we looked at 3 different brain regions. And what we wanted to do was to look at individuals with Parkinson's disease, but compare people with and without different symptoms. So, for example, we're pretty interested in dementia, but also other symptoms as well, such as aggression, depression, and anxiety, for example. For this was actually data that was given to us very kindly by a collaborator, Professor Nigel Williams at the University of Cardiff. And they hadn't actually performed it had been used as part of an MQTL study, but they hadn't actually used it for an EWAS because they actually only had about 10 controls and 90 AD, but they allowed us to then use their 90 PD samples to be able to look at these disease endophenotypes. So essentially what we did was we did our weighted gene correlation network analysis. So these are each circle on here is a different module. I presented this in a slightly different way because we're obviously looking at multiple brain regions. And essentially anything to the right of the dashed line is Bonferroni significant. And you can see that in 2 brain regions, so the substantia nigra and the caudate nucleus, we're seeing 2 Bonferroni significant modules. They're different modules, one associated with depression and one associated with aggression. When I was talking about the spread of disease, the substantia nigra is where we see disease earliest, and then into the caudate and then the prefrontal cortex later. So then we wanted to focus on these 2 modules in a bit more detail. So this is so gene ontology analysis of the 2 modules. And you see the substantia nigra depression module, we're seeing an association of lots of pathways that are associated with synaptic processes, whilst in the caudate nucleus aggression module, we're seeing an association with lots of things perhaps associated more with mitochondria and energy generation. We then went on to do our cell type enrichment analysis, and this is just looking at the module associated with depression, the substantia nigra, and what we saw was an enrichment of the module in excitatory neurons, inhibitory neurons, and dopaminergic neurons, which we thought was quite interesting. Following on from this, we've recently been doing an EWAS of Lewy body diseases. So for this, we've been doing 2 different EWASs. One's been looking at neuropathology. So what we've been looking at is around 270 samples, looking at 2 different brain regions. So the caudate, the cingulate gyrus, which affected relatively early in disease, and the prefrontal cortex, which affected relatively late disease, and looking for an association of DNA methylation with Lewy body Braak stage, very similar to what we've been doing in Alzheimer's disease. And our second study, which I'll touch on in a minute, was a clinical study, where actually we're looking at a difference between control, DLB, and PDD to try and identify perhaps specific loci that are associated with those different disease endophenotypes we'd identified. And just to say, one of the issues with doing any kind of study on post-mortem tissue is that often we have coexisting AD pathology, which I mentioned earlier. And so for this study, what we've done to try and get around this is we do have some AD pathology. We've made sure that's balanced across the group. So when I say control, some of those controls will have AD pathology to match the pathology we're seeing in the other two groups. So this is the clinical EWAS. And so this is looking at a difference between the two Lewy body disease types. Oh, sorry, I was meant to come up with something else. Essentially, what I wanted to highlight was that one of the genes we've seen here has been associated quite a lot in different Parkinson's disease, EWASs and GWAS studies before. In the neuropathology EWAS, when we formed it, so we've got quite a lot of samples here, and we've meta-analysed the two brain regions, but we actually only saw one genome-wide significant loci, which was this one shown above the red line. This is it shown here, PTPRN2, and again, like I said earlier, for the clinical EWAS, actually this one has also been associated quite heavily before with different Parkinson's phenotypes. What we then wanted to do was see if we could try and meta-analyse this. So we managed to access other EWASs that are being performed by collaborators in the prefrontal cortex with DLB pathology. And so we've meta-analysed now 758 unique individuals, some of those with two brain regions. And when we've done this, we've actually been able to identify 7 Bonferroni significant loci. And all of these show consistent direction effect across the 3 cohorts. So shown here is a forest plot, but you can see across the 3 cohorts we've got for this one example, we're seeing increased DNA methylation across the 3 cohorts. What we've then gone on to try and do was to try and look to see whether the signatures we've previously identified of AD, which I talked about earlier, so those 220 loci, and the ones we'd identified here, whether actually those are specific to each neurodegenerative disease or perhaps they're common to neurodegenerative disease. So shown on the left is where we've basically correlated the effect size of our the Lewy body, the effect size we see in the Lewy body EWAS with the effect size in the AD EWAS I talked about earlier for the 220 loci we'd identified and published a few years ago in 2021. And we can see there is a correlation. So we're seeing those 220 loci we're also seeing in DLB. And similarly, when we do the same with our DLB loci, we're also seeing the same. So the DLB loci are also being seen in Alzheimer's disease, but they're not the same top loci. When we then do a cell type enrichment analysis, we can see that the genes that we'd identified in our previous AD EWAS meta-analysis, they're predominantly enriched in microglia and astrocytes, which matched what we'd seen earlier. It was in glia subtypes. But actually, the ones we're seeing in DLB seem to be enriched in pericytes specifically. So it seems to be slightly different cell types that are being affected in the two diseases. And even though we're seeing common differences between the two, the most significant ones we're identifying are specific to the two disease types. And then another EWAS that we've recently done is we've looked at Huntington's disease. So for this is quite a small cohort. We've only been looking at 20 HD and around 20 control. We've been looking in 3 brain regions, so the striatum, cortex, and cerebellum, and we basically performed an EWAS across these. The striatum's affected early in disease, which is why we've used that, why we're seeing so many changes there. And then the entorhinal cortex affected in the middle stage of disease. Cerebellum is, although you do see movement changes, you won't necessarily see the pathology so much in the cerebellum. This just then shows that our top loci, one of the top loci we'd identified, we actually found in our regional analysis, so PTGDS, and we see that in the replication cohort, which is the blue and red, shown on the bottom, where we're using instead pyrosequencing, we again see a consistent change across this region when we look at multiple sites. And you can see on the far right that in Huntington's disease, which is blue compared to the controls in red, we see consistently decreased methylation across this region. And actually, we've gone on to perform this pyrosequencing assay for this gene across a number of different dementias and neurodegenerative diseases. And although we see it in Huntington's disease, we only really see it to a tiny extent in the entorhinal cortex in DLB and Parkinson's disease, but not in the other dementias. Following on from this, we've recently been funded by the MRC, which is the Medical Research Council in the UK, to do a big EWAS of Huntington's disease. So we're going to be looking at 400 samples. And actually, as part of that, we're going to be using long-read sequencing, which will allow us to look at methylation within the exon 1 of the CAG repeat, which will be really, really interesting because it will allow us to do allele-specific methylation changes as well. We've also been doing some work in blood, so most of our work's been in brain, but we recently published a couple of studies which have been an EWAS looking at blood. So this, we looked at 900 samples in the EMIF-AD cohort, and what we did was an EWAS looking at the association of DNA methylation with 15 different CSF biomarker measures, essentially try and identify perhaps methylation changes that could be important biomarkers, or at least tell us something about the biology in the blood. We identified, perhaps unsurprisingly, because Alzheimer's disease is a disease of the brain, we didn't identify many changes, but we did see some with association for neurofilament light and YKL40. The one that I just wanted to really focus on is the YKL40 one. So shown in A on the top left is the Manhattan plot. And you can see, we see these 5 genome-wide significant loci in YKL40, which are actually within the YKL40 gene, which suggests it's an MQTL. We then went on to look at this in a bit more detail. So the little bar plots shown in D, you can see that it's clearly an MQTL, and you see based on the genotype, you see different amounts of methylation for the two of the sites just shown here, and actually a really nice correlation of the methylation, which is shown on the x-axis in the far right one, with actually the amount of CSF YKL40 as well, which then we show again at the bottom. And actually what we did was we went on to do a causal inference test, which actually suggests that, because YKL40 is a previous GWAS hit as well, that DNA methylation actually mediates the association between the genetic variation and CSF YKL40 levels. Obviously, we're measuring DNA methylation in the blood, so that's a hypothesis, but we suggest that if we were to look at YKL40 in the CSF, we could actually show that the genetic variation in YKL40, which we know is associated with Alzheimer's disease and causes change in CSF levels, is driven by methylation changes. We've also gone on, just to briefly highlight, we've gone on to do a WGCNA, mainly because we had 15 different CSF biomarker levels, so we wanted to see if there were any modules which are associated with multiple different CSF biomarkers. And these are 2 of the ones we identified. So, for example, the orange we can see is associated, we see a module that's significantly associated with A-beta levels in the blood. And we can also see another one which is associated not just with A-beta, but also with levels of tau, which is the Saddle-Brown module. And these are the gene ontology analysis, and you can see that one of these is particularly associated, the orange one, with synaptic processes, and the other is more associated with cellular development. And then finally, I just want to finish up by talking about some microRNA sequencing. So most of our research data has been on DNA methylation. But in the last few years, we've also got interested in doing looking at other epigenetic mechanisms. So microRNAs are another epigenetic process that can regulate gene expression. We know that there's been several candidate studies that have identified microRNAs, which we know are really robustly associated with Alzheimer's disease. But what we wanted to do was, and there have been some genome-wide studies which have been done on microarrays, but what we wanted to do was use RNA sequencing to do an unbiased analysis of microRNAs in Alzheimer's disease. And we started this off as a pilot study, and then we ended up adding more and more samples. And actually, in the end, we've managed to accumulate data from over 900 samples, and we've managed to meta-analyze these. We've then gone on to do QTL analysis, like I described earlier, and look at causal inference tests. But also, what we've done for a lot of these cohorts is, as well as the small RNA sequencing data to analyze microRNAs, we've also generated matched RNA sequencing data so we can directly look at the change on gene expression, and we've done mass spec as well so we can look at proteomics too. The RNA sequencing was only in 3 of the cohorts and the proteomics in 1, but essentially we can infer some relationships from that. So this is a volcano plot of the microRNAs. So you can see everything above the dotted line was genome-wide significant. We've actually color-coded this, so based on the matched gene expression and proteomic data, if we then saw a significant correlation with the mRNA, it's shown as a box, and if it's with the protein, it's shown as a circle. If it's with both, it's a triangle. So you're seeing highlighted here the triangles, which are the ones where we're seeing a genome-wide significant change in the microRNA in Alzheimer's disease, but we're also seeing a significant correlation between that microRNA and its target mRNA and downstream protein as well. So this is essentially showing for those microRNAs that we identified where we're seeing an effect on mRNA shown on the top of the circos and the protein shown on the right of the circos. And this is also just to highlight that microRNAs are a really interesting study, but also quite difficult study as well, because they have multiple targets. So there'll be multiple mRNAs that are regulated by microRNA, and so it makes it actually quite difficult as well to work out what the biological relationships are here. So as I said, we then took our microRNAs and we took their mRNA targets and we looked at the cell types they're enriched in. What we saw, which is quite interesting, is the down-regulated microRNAs, which are shown on the left here. We're seeing an enrichment in, for example, excitatory neurons, which you can see here as this big yellow bar, whilst for the upregulated microRNAs, those seem to be more enriched in gliotypes. So we're seeing significant enrichment in microglia and oligodendrocytes. Just taking our top microRNA, so microRNA 1323P, one of its gene targets, so you can see here the low Braak stage of the controls compared to the high Braak stage of the AD. We can see a significant difference in microRNA expression, so reduced levels. We then see in the same samples increased levels of the target mRNA, qDPR, and also increased levels of the protein as well. And what was quite interesting is when we then looked at the correlation, we can see there was a significant correlation of the protein with the microRNA, only in the AD samples, but not in the control samples, which suggests other gene regulatory mechanisms might be important. We're currently including circular RNAs and long non-coding RNAs in our analysis. Those are upstream regulators of microRNAs. And we also have gone on to use basically microRNA mimetics. So we've basically been able to artificially add the microRNAs to neuronal iPSCs and microglial iPSCs. And we can see that actually when we add the microRNA to the cultures, we see a decrease in the expression of both transcript variants of the qDPR gene. Another really interesting thing with this is as I mentioned, we went on to do some MQTL analysis to see whether there was any microRNAs which were driven by genetic variation. And based on our methylation changes, we thought we'd probably find loads. Actually, we only found one interesting microRNA, QTL, and that was actually between our microRNA 132, I think it is, and ApoE, which is really interesting because actually they're on different chromosomes, which suggests that microRNA 132, which we know is one of the strongest hits in Alzheimer's disease, it's been talked about in candidate studies for years, that is also associated with ApoE, which we know is the strongest genetic association with Alzheimer's disease, and it suggests potentially they're interacting in 3D space, which is how they're looking at that. So we're trying to, at the moment, do some 4C to try and look at 3D interactions within the genome. So in summary, we've identified a number of genes that are epigenetically altered in AD and other dementias. Meta-analyses, and I think these are going to become more and more popular in the literature as well, giving us the power to identify robust and reproducible changes. The microRNAs and now the integrative analysis, where we're looking at circular RNAs, long non-coding RNAs as well, are an area we're really interested in. There are limitations, however, so this is probably every reviewer's comment I got on every grant and paper. Unlike GWAS, we don't know if an EWAS gene is causal in disease or secondary to disease process or even medications. One of my PhD students who's just finished, actually their PhD was focused on doing epigenetic editing, and so we were taking the ANK1 gene and trying to alter methylation. It technically worked. We could see about 40 to 50% change in methylation, which is great. What we saw was massive off-target effects using the CRISPR system. So actually, we can't really infer anything about the downstream biology because it's probably working across the genome and it needs more refinement, those methods. A lot of the stuff I've talked about today is bulk tissue or cell population level change and not single cells, and single cell methylation sequencing is really expensive still. We are doing more and more studies. So the Huntington study I mentioned that we're just starting up, that's going to be in cell-sorted populations as well. There's other factors that affect the studies, heterogeneity, the fact we're looking at post-mortem tissue, maybe that's okay for methylation, but for gene expression, your post-mortem delay can really affect the tissue quality. And also that we're doing more and more long-read sequencing, not just actually of the genotype and to look at methylation as well. We're also doing long-read sequencing of RNA as well to identify novel transcript isoforms too. So just to acknowledge, those are my groups. So I've talked about a whole range of different studies done by different people, but they don't let me in the lab anymore. So this is completely a teamwork by some amazing postdocs and PhD students with me, and also the wider team at the University of Exeter. And also, when you do meta-analyses, you really need to rely on collaborators. So we're really lucky. We've got a really great range of collaborators who give us our data and work with us. So thank you.
Thanks a lot, Katie, for a very interesting talk. So any questions from the audience? Yes, please, Michael.
Yeah, thank you for a very interesting talk. I have 3 quick questions. One is when you look at the epigenomic profile or even the epigenetic level of those usual suspects, do you see a correlation with different stage of the disease? This is my first question. Second one, let me finish all in one go. And when you look at the microRNA and other markers from the CSF, do you also see them in the blood as well, whether there's a correlation? And thirdly, can you comment on these recent tests for the Alzheimer's, like Roche's protein-based tests? For example, looking at the A-beta 41 tau, p-tau, and APOE gene, and how do those compare with this epigenomic study, and how will they work together for example, better early screening of the Alzheimer's? And
I feel like I've just had a memory test. First question, the usual candidates, yes, we do see an association with disease. So for most of what we look at, it's BRAC as a measure of tau pathology, and we do see a nice linear relationship. Obviously, tau doesn't spread in a linear way. So the fact we're seeing a linear relationship, perhaps we could model it in a slightly different way, but we do see an increase over disease. For only one of the cohorts, we've got access to amyloid measures, and again, we see the same, but probably because when you've got increased amyloid, you see increased tau. In terms of the blood, that's really interesting. One thing I didn't touch on in the discovery cohort I talked about very initially was we actually did have blood available for that cohort. We saw absolutely nothing. We then published a few years ago an EWAS in a cohort of blood samples, around 350. And we just saw some changes, but again, nothing overlapped whatsoever with the brain samples. And it was only the most recent one where we've actually had the power of around 900 individuals where we're seeing changes. So methylation changes in the blood are very subtle. I don't think our methylation changes, even though we'd hoped it would be useful as a biomarker, I don't think that's probably its utility. But if it can tell us something about the mechanism, so for example, that's what's mediating the YKL40 genetic effect on CSF, then that's really important, I think, because perhaps targeting that upstream of the CSF, of the protein changes, where we need to be looking at changes. And then the final question, my memory's gone.
The protein-based tests.
Yes.
Yeah, the tau and p-tau, A-beta, apogene. I mean, Roche recently has an FDA-approved test, right?
So for the CSF, so the EMIF-AD cohort we'd used actually was a big cross-European study. So we did methylation, but actually partners within it also did protein measures. And a lot of those actually fed into some of the later studies that have led to the biomarker stuff. So there is actually matched protein data on these samples as well. So I think these tests are accurate. It's just about the sensitivity levels of them, so how accurate they're going to be for early diagnosis. And I think that's been the issue with why we've been so unsuccessful at treating the disease, is people have got pathology years before you're getting clinical symptoms. Probably the best thing we can do is PET scanning, but we just can't afford to do that on a population-based level. Thank you.
Any other question? aging and dementia is an important topic that affects more and more people. So we may have more questions. Yes, please.
Thanks for the fantastic, interesting talk. So I have two questions. So one is, all throughout you've seen that the cerebellum is not changed. It doesn't show the changes in epigenetic signatures as compared to other brain regions like entorhinal cortex. That's great. Can you illuminate a little bit more on that? Is there some simple explanation as to, okay, some genes are, some enzymes, for methylation is not expressed, so that's kind of simple. Or it tells us a little bit more about somehow the vulnerability of these brain regions, right? So that's cool. So then the question is like, can we flip the argument and kind of ask, how can we make the entorhinal cortex more like the cerebellum and kind of like reduce that kind of vulnerability, protect it a little bit more from AD, for example? So that's my first question. And second question is kind of simple, which is, have you, or do you have any plans to look at the patients who are receiving treatments, let's say the antibody treatments against A-beta, and look at these signatures, compare them with or without treatment?
Yeah,
Thanks.
so I'll answer the second question first. So yes, it'd be great to, but it's kind of accessing those kind of samples sometimes is quite difficult because they're part of clinical trials. We've been really fortunate. The very first amyloid immunotherapy clinical trial, which was the ELAM one in the early 2000s, we've got access to 20 post-mortem brain samples from people who are on active AD immunotherapy. And so we've been able to look at the association of amyloid clearance and the amount that they'd seen post-mortem, the amount of microglial activation with DNA methylation. And we've used the WGCNA approach to identify modules specifically associated with the clearance of amyloid. I didn't show it today because it's kind of just being finished up, so kind of didn't want to give the wrong story. In terms of cerebellum, it's really interesting, actually. I think you're right. We need to think about this in the reverse, not what's causing disease, but actually far more positively, what makes a neuron resistant to pathology. The fact that most of the AD changes we're seeing are driven by glia, though, suggests to me that perhaps it's something that's specific to the way glia are responding in different brain regions rather than perhaps necessarily the neuronal susceptibility. There's neuronal differences. For example, you have pyramidal neurons, which you don't see in perhaps the other brain regions, but we know there's neuronal diversity within the cortex itself anyway. So to me, it suggests it's something that's glia specific, perhaps. I suppose I'd hope that what we're seeing now is more and more single-cell RNA sequencing studies being done across multiple regions of the brain in diseases such as Alzheimer's disease, and I think those will be the ones that will probably be able to answer exactly that question. So I think probably not our research, but I think that's something that will definitely be coming out in the field.
Hi, Katie. Great talk and very important work. So my question, I think maybe you've already mentioned it during your talk, but I didn't quite catch it, is that so for the meta-analysis of the EWAS study, do you also have the GWAS data? So you know that like the patients with the top EWAS hits actually are either high or low with those association with those quite well-known genetic alleles, variants?
Yeah.
So meaning that like the epigenetics is more driven in those patients without a clear epigenetic variance, genetic variance?
It's a really good question. So all of our studies have just focused on EWAS, but you're completely right. So we have recently generated matched GWAS for all of the samples as well. Part of that is we want to do an MQTL analysis. Obviously, there's brain MQTL studies that are out there, and what we want to do is doing one for cerebellum versus cortex. No one's published that before, but also AD specifically. The other thing as well to do is look at the genetic effects, like you're saying, high versus low. We just started a study. So one of my PhD students, their PhD is looking at ApoE, and basically they're doing an ApoE EWAS specifically to look at that, just because ApoE is kind of the easiest because it's got the largest effect size and we're looking at a single gene.
Thank you. So I think, I think we're very honored and happy to have the chance that Katie can be with us today. I think speaking first on this topic of epigenetics in dementia, but broader than that, we are also trying to establish collaborations between City University of Hong Kong and University of Exeter. We have already got very good discussions and communications, and I think we have had teams visiting each other. So I think all of your interest and kind of collaborations from researchers level would facilitate that and make that bigger success. And Hong Kong and also City University of Hong Kong is pushing very hard on many of these life and health technology related area. So with the Institute of Digital Medicine, Professor Michael Yang, our Senior Vice President on Innovation and Entrepreneurship was just here. And we also are working hard on collaborating, establishing collaborations to submit a proposal to the Hong Kong government on some big research institute that I think there's a lot of room for collaborations as well. And we'll talk more about that. So with that, please join me to thank Katie once again for her time and for sharing the exciting insights from her work. And we look forward to continuing this dialogue. Hopefully this is just the beginning of many, many successful exchanges between us and Katie and her colleagues in Exeter. So thank you, Katie.
Thank you for having me.